welcome back to the lecture series in nanotechnology in agriculture. So, we were discussing about iron pyrite as a seed biostimulant. So, next couple of classes we will be discussing about the mechanism of action by virtue of which iron pyrite helps in enhancing the production of the crops. So, as of now it has been clear to most of you that iron pyrite as a complete molecule is not entering most likely inside the seed. What is happening is that since it is insoluble in water, it is forming trace amount of hydrogen peroxide, iron in +2 and +3 state, and SO4 minus ionic species. So, there are cations and the anions and a peroxide. It is kind of a milieu, and all these rest of these amounts, all these cationic and anionic species plus the peroxide is in a very trace amount, and whatever is happening, it is happening because of these ionic milieu. But then in my previous class I introduced the concept of why a seed is not allowed to germinate. I told you that the germination is being prevented by a series of antioxidant molecules which are present there, and as you know, in the biological system there are 3 key antioxidant molecules: catalase, peroxidase and superoxide dismutase. The nature has equipped us to live in an oxygen environment by virtue of these antioxidants. And here just I will take a slight detour to tell you oxygen molecule can come very handy in your immune system, where you have to destroy the foreign particles or foreign cells. The most of the macrophages present in our body, they eject out your free radicals in order to destroy XYZ, but at the same time these free radical can affect your system also by destroying cells. So, it is kind of a double-edged sword. So, somewhere in between we have drawn this line of having a series of antioxidant enzymes which ensures there is a balance for oxidant and antioxidant kind of is being maintained. Okay. So, resuming, so there are 2 concepts what we introduce, and now today we are going to put all of them together. Okay. So, just a small recap. I talked to you about the chickpeas, where you saw how in the water post treatment the growth of the chickpeas post FES2 treatment is much more higher. We talked about the increased germination percentage, enhanced root growth, enhanced shoot growth and overall biomass increase. Then we talked about the field trials, and in the field trial I talked to you about the spinach seeds, and again for 12 hours treatment in water and correspondingly in water plus iron pyrite. Post that these seeds were sown, maintaining the same nutrient status for the soil, same amount of sunlight, same amount of water, no fertilizer and equal amount of organic matter whatsoever is present in the soil. And what was observed? There is an increase in the leaf size, there is an increase in the biomass, there is an increase in chlorophyll, increase in manganese, sulfur, calcium. Now, where from these effects are coming? So, this is where we stopped. I told you that a dormant seed remains dormant because there is a battery of antioxidant enzymes which prevents or prolongs its dormancy. So, to use the right phrase here, it prolongs the dormancy. Now, how it prolongs the dormancy and where pyrite acts onto it, how it prolongs the dormancy. But before we get into that part, that how it prolongs the dormancy, let's again summarize. So, you are having pyrite, you are having trace amount of H2O2, trace amount of 3 plus, you are having trace amount of SO4 2 minus, and you are having trace amounts of H plus ions, and of course, you are having water. And I told you a seed, when you look at a seed in its dry dormant state has a battery of antioxidant enzyme, and a seed has a huge amount of a strong reserve of carbohydrate, and this is this carbohydrate which decides or is the first source of energy. Now, this carbohydrate, when seed is germinated, it is the water molecules which comes and it activates its enzymatic machinery around, and this carbohydrate is broken down to generate energy. Now, what is breaking down the carbohydrate? The enzyme, which is, so those of you have forgotten, carbohydrates are long chain of glucose, galactose, mannose, and all sorts of things. They could be starch, extremely long chain, they are crisscross, and there are a lot of these, if you just recollect, there are a lot of these moieties forming something like this. Okay, this circle is representing each one of those glucose and moieties and like so on and so forth. So, these are crisscross chains like this, long chains, and this is what starch and all these molecules are. So, now so this is essentially a big carbohydrate molecule. Now, in order to break a carbohydrate molecule, what one has to do is, so the carbohydrate, each one of these individual units, what you see, these are the monomers. These monomers are attached or form a polymerized structure through a bond called glycosidic bond, which is this one. Now, in order to break carbohydrate, for it to release energy, what one has to do is one has to break up this bond. If you can break up this bond, then this will release energy, and this is precisely the energy we are talking about. But breaking this bond is not an easy job. There are specific enzymes which does that job, and one of the key class of enzyme which does that job is called amylase. So, amylase activity, when the seed is from its dormancy is heading towards germination, what we observed is amylase activity goes up significantly. because of the amylase activity going up significantly, it leads to the breakdown of starch into small molecules like glucose. This is your glucon D almost. So, basically you want to go for a 100-meter dash, take a glucon D and you run. This is precisely how a seed germinates in an environment which is extremely challenging for it to grow. It is not easy. Now, these producing sugars which are getting produced, they are the ones which are source of energy. Now, if you think the way we can hasten this process or make the germination to happen is some way or other, if we could ensure the amylase activity is initiated. Now, what is the role of these antioxidant enzymes? So, the antioxidant enzymes prevents the amylase activity. That is what it does by stopping the amylase to act upon. So, what we observe is amylase activity which is responsible for breaking down of the big starch molecules is being stopped by these antioxidant enzymes. So, this is how nature maintains the balance, but the very moment water comes and started activating this zone, the amylase gets into the play. But now, this is all fine, so far so good. Then, what makes the presence of pyrite so very important? So, if you look at it here, chlorophyll and of course, the germination percentage, how pyrite is playing a role in germination. Now, look, if you look previously also the same thing, germination percentage. So, how this material is acting? Now, this is like, I have to give a situation. I am breaking a starch, you are breaking a starch. But if I break a starch faster than you, then I will be harvesting more energy faster than you. Okay? So, say for example, you have A and you have B. So, starch, so our test is starch breaking speed. Okay. So, if the starch breaking speed of S, if I represent it by S, and SB is the starch breaking speed of SB, if SB is more than SA, then SB will germinate faster and more successfully. Okay. Faster germination and more percentage germination and more successful germination. Okay. Now, is FeS2 playing such a role? Now, having said this, I will come back and visit this molecule now. Now, if you look at this, if you remember one old reaction called Fenton reaction, where in the presence of iron you produce hydrogen peroxide, a very old chemistry. I will request you to look at Fenton reaction. Now, if you look at the Fenton reaction and if you look on this red alphabets here or red words here, something very similar to Fenton reaction, production of H2O2 in the presence of iron. So, in other words, FeS2 chemistry or aqueous chemistry of FeS2. So, let me put it aqueous chemistry of FeS2 is similar to or very close to Fenton reagent or Fenton chemistry. And having said this, let me highlight one point here. For those who are interested in degradation of organic waste which are carcinogenic, which are extremely environmentally hazardous, there are places in the world where they use large amount of pyrites to execute those breaking of those huge organic molecules. How they do so? Now, here how they do so. You have these hydrogen peroxide here. Peroxide can act. This was way back in 1920s. There was a wonderful paper in Journal of Biological Chemistry which was published. It said that hydrogen peroxide can mimic, mark my word carefully, can mimic the activity of amylase. So, in other words, FeS2 or pyrite is mimicking, this whole thing is FeS2 plus water is similar to amylase. Okay. So, in other words, one can consider FeS2 plus water system as equivalent to amylase or artificial starch breaking enzyme system. Okay. So, now let me get back to that example what I gave you here. I am just underlining this in pink. Now, you have A and B, and B has higher starch breaking speed. So, automatically B will be performing faster and will be more successful. That is precisely what is happening when FeS2 in the presence of water is generating this trace amounts of hydrogen peroxide, which is mimicking amylase activity and going there, and what it is doing is it is chopping off these glycosidic linkages and generating energy faster. So, it is generating more, reducing sugar far more faster than naturally or in a normal process amylase is doing, and by the process it is ensuring a faster germination, and having faster germination leads to a faster growth, and in that whole process the plant is getting, as we will proceed further, is getting a support from SO42 minus, Fe2, Fe2 plus, Fe3 plus. That will be discussing subsequent classes, but there is one more thing which happens. These hydrogen peroxide, apart from it, possibly activates a pathway which is called brassinosteroid pathway. Brucinous steroid pathway, which promotes a faster growth. And lastly, H2O2 itself is a signaling metabolites for growth and development. So, hydrogen peroxide by itself does multitude of things, but the problem and the challenge is that you can argue that then why don't we put hydrogen peroxide directly? The challenge is it acts in such a trace amount that it is not really one of the translatable technologies. Second, there are certain very unique role, as we will diverge it further, you will observe of these released SO4 minus, Fe2 plus, Fe3 plus, and this milieu of slight acidic environment. As we will proceed further, we will describe those, how they play a role. So, to conclude, iron pyrite mimics in the presence of water the Fenton chemistry, point 1. This is this point. Okay. It, while mimicking Fenton chemistry, generates hydrogen peroxide, which mimics amylase activity, and just like amylase, it breaks down or chop off the glycosidic linkage, and thereby increasing the possibility of the plant to absorb more or get more energy in the shortest possible time. Because you always have to realize, when you put a seed in the soil, it is facing one of the harshest environment you can think of. There are microbes, there are competing seeds, there are everything all over the place is opposing it. So, it has to grow faster, it has to emerge out from the soil. So, in order to do that, it has to really do it real quick, real fast, and that's precisely this process is facilitating without even getting into it. And then it possibly activates the Brucinous steroid pathways, and in itself it acts as a signaling molecule for development. And as we will proceed further, we will talk more of how FeS2 affects the root geometry, which has a profound impact on nutrient deficient soil. And further, FeS2 water system is an artificial starch breaking enzyme system. Okay. So, we will continue this in our next class. Thank you.